Hello everyone. In today's video, we are going to explore molecular docking using that is one of the most widely used computational tool in modern drug discovery. Okay, but before we jump into the practical demonstration, let's first understand the concept of molecular docking and why it has become such an important steps in the field of computer-aided drug designing or in silico drug designing or computational biology, right? So molecular docking is a computational technique that is in a computational method that predicts the preferred orientation of a small molecule, right? Commonly referred to as a like a ligand, like a compound, like a small molecule, right? So when a ligand when a ligand binds to a target protein, that is a protein molecule that we can call macromolecule, and it could be receptor, it could be enzyme, it could be protein, right? So that called the target protein and that target molecule. Okay, so the main goal of docking is to estimate how strongly and how specifically a compound, a ligand, interacts with its target protein, right, with their residues, with their active site residues. In simple words, it helps us to answer the question like, will this ligand molecule fit into the active site of the protein? How stable is this binding, right? Protein ligand binding. Could this molecule potentially act as a drug? And the thing is that drug discovery is a long, expensive, and resource intensive process, right? So traditionally methods involve screening of thousands of millions of molecules, right? Experiment, so which takes a lot of time. So this time we can reduce via this in silico drug designing or computational drug designing. So molecular docking is just the scoring function of and to generate poses, generate the conformations of best poses and conformation of ligand and protein molecule to fit their with their pocket of the protein molecule, right? And we can have a dock pocket. So this is all about the docking. You can see in this animation how small molecules in various colors, right? You can see how this ligand molecules bind and interact with the protein molecule, right? So this is all about the molecular docking, how the ligand molecule binds with target protein. Okay, so the nuclein 3A derivatives, they are just the small molecule. I have written this, and the in the brown and gray color in the space, that is the protein or target protein, right? So this is something like this. This is the thing where the thing happens in molecular docking. How the small molecule, how the ligand molecule, how the compounds and docked with the binds with the protein, and this thing called molecular docking, right? So so having understood the theoretical background of molecular docking and its importance in structure based drug designing, we now move towards the practical implementation of molecular docking using AutoDock Vina. So in this demonstration, we will perform the molecular docking on a co-crystallized protein ligand complex obtained from the RCSB protein data bank. Okay, so the selected PTB ID is 4G7, which represent the MDM2 protein. Okay, and this MDM2 protein structure we will be used to define our docking grid parameters and evaluate the docking accuracy. Okay, so let's get start the molecular docking using Autodock Vina. Okay, so we are in our current directory. Okay, this is molecular docking, and all the files, all the input files, the structure files, and the docking script uh, we have in our current working directory. Okay, this is the script, and this is the parameter file, and this all are the ligand molecules I have designed for the molecular docking against this target protein. This is the MDM2 protein. Okay, And yeah, but before that, I'll tell you step by step how can we proceed with this protein for the molecular docking. Okay, so first I'll tell you what is the 4HG7, what is the MDM2 protein. Okay, this is MDM2 protein. Okay, 4HG7. This is the unique PDB ID. Okay, there is some internet issue. Uh, yeah, yeah. There is the protein crystal structure of MDM2. There is the complex with nuclein 3A. This is the ligand molecule. Okay, and yeah, this is the protein all data, and we can download these structure files via download files and go to the legacy PDB format. Okay, but I have already downloaded this file in our current working directory. This is the file. Yeah, you can see there's the ligand. Yeah, this the red one is a color, this red color molecule is ligand molecule, in the blue color this is the protein molecule. Okay, there are some water molecules presented and ions. So yeah, uh, now the thing is that we have to prepare the structure, right? So the structure preparation, we can use Pymol and Discover Studio Visualizer and Chimera as well, and the MGL tool, Autodock MGL tool. We are just going to use in our case. Okay, so just we have to open our AutoDock Vina MGL tool. Yeah, this is the interface of the Autodock tools. So first we have to import the structure via reading the molecule. This is the 4HG7 molecule complex, and yeah, you can see this all the protein molecule, and this is the ligand molecule. Okay, I'll show you. Yeah, is the nuclear molecule I have already removed from this structure. Okay, so just we have to prepare the structure via removing all the non-residues part. Okay, so just go for edit and delete photo molecules, and add charges, add Coleman charges, then go to edit again, go to hydrogens, add, and add polar hydrogen only, and just go to edit again, miscellaneous, and just go for check for the missing atoms. If there is some missing residues are there in the protein molecule, so target protein, so if there are missing residues available, so we will have to just repair them and add the missing atoms. Okay, so yeah, you can see there is a zero residue with missing atoms, so we'll have to just dismiss this thing. Okay, yeah, so then go for grid, macromolecule, choose, select molecule. 4HG7 already have per atom partial charges. Do we want to preserve these charges instead of adding extra charges? Yes. So yeah, Yeah, this molecule is saved in our current working directory. Okay, so just we have to cut it from this. Okay, so just now the second step is to prepare the ligand molecules. We have the prepare we have prepared the receptor molecule, and this is the receptor.pdbqt, and now we will have to prepare the structure ligand molecules. So as you can see, there is total 11 structure. Okay, so OB minimize, and this force field will utilize in this in our case. Okay, so the ligand preparation will done by the open bevel. Okay, O bevel minus I pdb dot s pdb output O pdbqt minus O dot pdbqt. Okay, yeah, so as you can see, this is the open bevel flag for the open bevel. IPDB, this is the input PDBs, these are the PDBs it will utilize, and star PDB also, and output is PDBQT format, because the AutoDock Vina will only proceed with PDBQT prepared files. Okay, so the ligand molecule and the protein molecule should be in the PDBQT format. Okay, so that's why we are just going for the conversion of the in sense of the preparation. Okay, so just go for the hit the enter button. Yeah, you can see there's the total 12 molecule converted. Okay, yeah, you can see there's total of the 11 molecules. Then just write the ls ligand dot txt. Okay, so yeah, we have the ligand.txt file, and in this file all the things we have to remove other than the only ligand molecules. Okay, so yeah, only the pdbqt files we have to keep in this ls.txt file. Okay, other things we have to remove other than this. Okay, so 1 2 3 4 5 6 7 8 9 10 11. Yeah, there's total 11 structure ligand molecules, and just we have to save it. Okay, after that just we have to write the final command, but before that I'll tell you what is the Perl script. Okay, I have designed modified this Perl script, okay, according to our easiness and ease, for our ease, and some parameters. Okay, so this is our Perl script for the molecular docking. Okay, just write Perl and hit the enter button and just go for ligand.txt file. And yeah, you can see the molecular docking is started using AutoDock Vina. Okay, so we will have to wait for some time to completely run the molecular docking, and all the results will save in one single file, and the second file will generate that will just the rank rank the complexes and hit molecules according to the docking score. Okay, so two molecules already have docked and the third molecule is under process. Okay, so we will have to some wait for some time. Yeah, we have one, two, three, four, five, six molecule we have docked, and the seventh molecule is under process. Yeah, it's already done, and some of molecules remaining. I have made the script in for the very easy. Okay, so if there is a lot of thing to run step by step, different kind of command lines, but I have merged them in a single modified Perl script. So using this script we can just we will have to go for the 4 and 5 command lines, and our molecular docking is already done. So yeah, almost we are there. Yeah, docking is completed for all the compounds. All docking log files save in final log.txt file, text file, and summary save in docking summary.txt file. Okay, high to low docking score. So I'll show you the results. Yeah, there is the output files. This is the output files AutoDock Vina generated. Okay, and the results Yeah, there's the results. You can see for the MLT10 molecule there is the minus 8.13, minus 8.14, and for MLT1 there's the minus 8.026, and similarly for all the compounds. Okay, and you can we can see the highest docking score, which compound have the highest docking score, and this docking score, this data is saved in the final docking summary file. Okay, yeah, you can see MLT6 have the minus 8.94 highest the docking score. this is the this is the compound MLT6. Okay, so yeah. So our molecular docking is completed, right? Yeah, so using pymol we will just yeah. So using pymol we will visualize the dot complexes. Okay, so first open the receptor file. Okay, so just go for receptor PDBQT. Yeah, there's the thing, there's the protein. Okay, and just go for open the dot complexes. Okay, so we have open the summary file. Okay, and there's the thing, the MLT6 molecule has the minus 8.94 docking score, highest docking score, and this is the hit molecule. So this structure we will see the conformations, the binding. Okay, so MLT6, this is the MLT6 out dot pdbqt. Okay, so yeah. just active select and preset. Okay, so you can see there is the poses, there is the different conformations of poses of the ligand molecule. Okay, so this is the first pose that has the highest docking score, right? So, and this is the second pose, third pose, fourth, fifth, sixth, seven, 8, 9, 10. Okay, so total 10 poses are there, but the in case of selection of which poses, so it will generally based on the highest docking score. Okay, so yeah, the first pose we will use for next or studies, right? So this is the best pose and highly uh, interacted with our residues, MDM2 residues. Okay, so just go for file, because this complex we have to save in as the model. Okay, the complex model. So just go for file, uh, export structure, export molecule, and just save it, and just keep the thing pdb. Okay, and just give the name mdm2 mlt6 complex. Okay, and just save it, and yeah, you can see that is the saved complex protein with our dog molecule. And yeah, you can see, I'll show you. Yeah, you can see there is the red color. This is our ligand molecule. This is docked ligand molecule. Okay, so yeah, this is all about the molecular docking. Okay, and this structure we will generally use for further in silico studies, computational studies. Okay, we can also visualize some residual interactions using different kind of visualizers. Okay, I'll show you this. Yeah, and just go for ligand interaction, and you can see the glycine 59 is interacted with this oxygen heteroatom as the hydrogen bonding. Okay, and these are the hydrophobic interactions: valine 93, histidine 96, isoleucine 99, leucine leucine 54, phenylalanine 55. So yeah, this is all about that. So this structure, after the molecular docking, we now have a well defined protein ligand complex which serve as the starting point for molecular docking, molecular dynamic simulation as the second most important method to analyze the stability of a protein ligand complex. So MD simulation is generally allow us to study how the complex protein ligand complex behaves over time under physiological conditions that provide insight into the stability, flexibility, and dynamic interactions. So by analyzing parameters such as RMSD, RMSF, hydrogen bonding,
and binding energy fluctuations, we can better understand the strength and the reliability of the docking process with this method. Okay, so if you want to explore in the detail, okay, in the much detail, I have dedicated videos demonstrating the MD simulation using GROMACS and open source molecular dynamics engine and suit, where I walk through the complete workflow of setting up the MD simulation and running and analyzing the protein ligand MD simulation. Okay, so you can watch my previous MD simulation video after the molecular docking. Okay, so all the things I have put in my YouTube channel. Okay, so how to install AutoDock Vina, and then how to dock this protein ligand using molecular docking using AutoDock Vina, and after that this complex structure. Okay, this complex structure, how can we utilize this complex structure for the molecular dynamic simulation. Okay, so then after that, how to analyze the trajectories, how to analyze their stability and dynamic behavior of the protein ligand complex, and finally how can we analyze the binding affinity, final binding energy, binding free energy to using MMPBSA and MMGBSA method. So all the things, all the major things are already there in my YouTube channel. So yeah, you can watch all the videos and learn based on these tutorials, and apply in your real-time research. Okay, so yeah, thank you.